that the best way to learn is to participate. Delia is a living kidney donor to her daughter, Marissa. Marissa Nahaney is a Squamish and Nisga'a Indigenous ambassador living in Eslahan, North Vancouver. She's a member of the Eagle Clan. Her focus is on carrying traditional teachings and protocol into modern world through her art, bringing lessons of humility, generosity, and respect. She's a founding member of the Sustainable Diversity Network at the David Suzuki Foundation, creating opportunities to learn about Indigenous Chen Shen Shui, elevating one another. Currently, she is working as the cultural coordinator and for language and culture affairs at Squamish Nation. Since 2011, Marissa Nahaney provides authenticity and creativity for all of your event needs, including graphic design, corporate gifting, and Indigenous talent. Nahaney makes it easy for you to deliver a memorable experience to your audience. So Marissa is also a patient partner with the CAN CKD Network Kidney Check Project. So I'll hand it over to them. Thank you.
Nuchaya Wenmahat, Dalgamha, Quansnot, Dilian Haney, Quincha, Aslahan Okomeo, Sko Umish Ni, Nishke Ni, Almanax and Squalin. I'll let Marissa introduce herself.
Tanoyab, Tanon Siaman Siaya, Mechem Koshaman, Marissa Mahani, Queensna, Skohotmesh, Ochomelch, Niska, Ochomelch, Ananax and Squalin Teat Seats. How's everyone following me so far? Translate, bonjour, comment ça va? No, I'm just kidding. Greetings to each and every single one of you here today. It is a true honor to be amongst all of you smart, brave people in the nephrology. My ancestral name is Mechoche. My English name is Marissa Nahaney, and I come from the Squamish and Nisga'a peoples. And as my mother, or as the bio said, my mother was my first kidney transplant back in 2011. And I was able to successfully live for 9 years with her transplant. And then 2 years ago, I received an anonymous kidney donor. And so it's without saying it, but I definitely really appreciate the work that you do. Thank you for dedicating your lives to something extremely important, because the kidney disease does not just affect myself, it affects my whole community. My mother worries about me. It's not a life to be living on dialysis. And my community worries about me, and they're always wondering what's going on. So when you're doing the hard work you're doing, you're not only saving a person, you're saving a community. And a lot of gas money. Those appointments and gas parking tickets get a little bit much.
I just want to welcome you all to the unceded ancestral territories of the Squamish, the Musqueam, and the Tsleil-Waututh. The lands in Vancouver. We have cared for these lands since time immemorial, and it's important for us to acknowledge that truth today as we meet and as we gather here today. Ocean.
Thank you, Delia and Marissa, for welcoming us today and for land acknowledgement and sharing your culture and bringing some positive energy and some levity to the space so we can focus on connections and collaborations.
Yes, I want to say thank you as well. It was very inspiring. Before we get started, I just want to make just quickly on a personal note, also acknowledge the fact my anonymous kidney donor from 5 years ago, 7 years ago, more than 7 years ago, who saved my life and whose name I will never know, but I'm eternally grateful wherever you are. Before we get started, just a few quick notes about today's meeting. This is a hybrid event. So please be mindful of both in-person and virtual participants. Speak up a bit, if you will, if you've got questions or something. Make sure that whatever microphones we have in here are loud enough that somebody at the other end of this network can also hear what you're saying. Photos will be taken throughout the meeting. So please let us know if you have any concerns. You don't want your picture taken? No? Or if you wish the photos not to be shared. I turn it back to David.
Thanks. So we have a pretty tight agenda today, so we're going to really try to keep on time and really reserve time for questions and discussion. So I'm sitting in the front row and I'll be watching you, and if I stand up, that's a sign. But there's some timekeeping in the back with Amanda. We'll have some guidance as well. And then I think one other important thing is this is a very diverse audience of clinicians, patient partners, researchers. So just make sure that we're being inclusive in our language so that everything's accessible. And there are lay summaries printed on tables for people to review as we go through the topics. Sorry, the slides keep. Okay. All right. Sorry, I'm not sure if my clicker is working. Okay. So this is the agenda for today. So we have a couple presentations starting at 9:30, going through Easi-Kidney with Adeera, SugarnSALT with Dr. Sridhar, the CARE-MRI trial with Thomas, and the PART study with John Gill, and then finishing up with Istvan Mucsi on patient-reported outcome measures, a pilot feasibility trial. Before all that exciting stuff, we're going to give us some updates about the CNTN and succession planning, and bringing some new blood in and passing along the torch. And then Karthik is also going to talk about the ACT, or Accelerating Clinical Trials Canada Consortium planning update. Karthik's unable to be here in person today, but he's presenting virtually. And then I'll be talking about one of the ACT or FA winners, which was me and my team for number 6. But there's been lots of other people within the nephrology community that have gotten successful funding too. So that being said, I'll hand it over to Karthik.
Great. Can you hear me okay?
Sorry, Karthik, I think I just skipped ahead. I'm going to hand it back to Hans, actually.
Okay. But I just want to check if you can hear me.
Yeah, we can hear you.
Wherever you are, this may not be necessary actually handing it back to me, because I think everybody knows who Karthik is. Anyway, I'm pleased to welcome Dr. Karthik Tennankore, who is joining us virtually today, as you heard. Karthik is the current co-chair of CNTN's executive committee. He is also a professor of medicine and the head of the Division of Nephrology at Dalhousie University. Karthik has been a long-standing leader in the kidney research, with a clinical and academic focus on improving outcomes for people on home dialysis and undergoing transplantation, both of which I qualify for. Today, he'll be sharing updates on CNTN's next phase, leadership succession planning, and key updates from the ACT Consortium. Please join me in welcoming Karthik Tennankore.
Thank you. Can you see me, or is it just audio? I prefer not to be seen.
Right now, it's just audio, but the people online might be able to see you.
Okay. So I actually don't have slides for this first section, but before I start, I just want to thank the presenters of the land acknowledgement. And I do want to say, obviously, I'm not able to be there with you, but I'm speaking to you from Mi'kma'ki, the ancestral and unceded territory of the Mi'kmaq people. And we recognize them as the past, present, and future caretakers of this land. This first part really is just meant to be an introduction. We haven't necessarily engaged on long conversations about, succession planning or CNTN 3.0, or I don't even know what number we're at, to be honest, at this point. But what I really want is just the opportunity to mention to everybody, I started in my bigger administrative roles in CNTN in 2019. And at the time when I took over as the co-chair of the executive, and I was co-chair of the scientific committee as well, which David is now, The terms of reference for CNTN were to have really a lot of movement in that kind of executive leadership, just to really give the opportunity for people to take part, be involved in the Canadian nephrology clinical trial community, and not just be part as members of committees, but as leaders of committees as well. So that was 6 years ago, and I'm in the same position right now. It's been a pretty interesting period. We obviously navigated, the pandemic and trying to make everything virtual for CNTN, and I think we did a pretty reasonable job of that. Navigated lots of changes in administrative leadership, co-leadership, and then most recently, which I'm going to be spending most of the time talking about, navigated our inclusion in the Accelerating Clinical Trials Consortium, which, as you'll see, has been, I'd say, mutually beneficial. Our members have done a lot for ACT, and ACT has done a lot for our members as well. But I do think it's time for change, and I think a lot of the co-chairs would probably agree, as would some members. It's nice to have some, fresh faces join the various levels of both our executive and our subcommittees as they exist right now. So, I mean, happy to open it up for discussion. I don't think we have a lot of time really to go over this, but it was mainly just, I'm not sure who's in the room, but to younger, more junior faculty, trainees, get involved. B, I think there are going to be some real good opportunities to take on leadership positions, and I'd strongly encourage, especially those who are getting their foray into trials. This is really going to be an opportunity to network, connect with your peers across Canada, but also with other networks and other groups, hoping for and assuming our ongoing connection with ACT as well. So I'll just take a pause. Maybe David, if you're able to just identify me, is there anyone else from sort of the administrative side of CANSOLV CKD that's in the room that wants to comment or provide any other thoughts?
Thanks, Karthik. I'll take a stab because I just want to make a comment. So I joined CNTN, back in the day of 2019, et cetera. So I was in the middle of my PhD at McMaster. And you provided mentorship that I was on the scientific operations committee. I was doing reviews, giving feedback, getting those skills, and developing as a trainee within Crescent and CANSOLV CKD. And now I get to work with Hans as scientific co-director. So I think there's a path for each trainee that's different. And I see people that are doing clinical trials and clinical research in the room that are trainees. So I'm just gonna, call out IO, Cal, Taiwan. There's lots of people here that are up and coming, and there's opportunities, and some already sit on, different chairs, different committees, whether it's the scientific operations committee with us or, the communications engagement committee or the capacity building. So there's lots of opportunity. Please come talk to us if there's questions about it. But that's my plug. It's really enjoyable work. It's really rewarding to be able to review and to give feedback. And I've benefited from the CNTN a lot of my career so far. So I don't know if there's anyone else from CANSOLV or Crescent or any other organization that wants to share anything. Just comment too that Omo is the new research coordinator for CNTN. She was unable to be here today because she's sick, but she's been fantastic. Please reach out to her or to myself or Hans for any of the scientific committee feedback or questions or how we can help you. But I want to acknowledge that this meeting wouldn't be possible without her today, and she did a lot of legwork into. But also the CANSOLV CKD team has also been really supportive and are here on the ground today, filling in and helping out. So thanks, Karthik. I'll hand it back to you.
Yeah, no, thanks, David. And just to sort of extend that further, there's probably going to be some, call outs for positions that are going to become available. In a staged fashion. We want to make sure that people who are there understand how things work. There's a little bit of overlap. I certainly will want to continue to be part of the CNTN because it's been such a rewarding experience for me and a learning experience as well. But I think we're going to work together as an executive to start identifying some succession planning and some strategies just to make sure that those who might be interested but are a little concerned about what's going to be involved, workload, that kind of thing, we can certainly navigate that. And I'll also say thank you to Omo, who, despite the illness, is still working even in the background quietly, helping me with sort of coordinating around slides and everything. So with that, we can probably move on to the next part, which is just an update on ACT. And I think, perfect. So I'm actually looking, I think, at the screen through the screen. So I'll do my best here to try to sort of go through the slides. But can we move to the next slide? So really what I want to do here and then open it up for questions and comments, and we have a little bit more time. Update on some of the ACT activities, Accelerating Clinical Trials Consortium activities in the last year that might be relevant to our network members, including some new stuff around contracts, regulatory, and RFAs. Highlight a little bit around the CNTN involvement in ACT. And David, if we get to that slide and you wanted to sort of expand further or do something, just let me know. A couple of calls to action, and then just touch a little bit on where ACT may be going in the future. And just so for those that maybe don't know, CNTN as a network was one of the first 28 networks that was invited to be part of the Accelerating Clinical Trials Consortium. Since that time, 6 other networks have joined. So there's 34 networks, in addition to a large number of other groups, including clinical trial units across Canada that are involved in ACT. And as you'll see, we've certainly done a lot and have been involved a lot in ACT. And I'm hoping that we can continue to be involved in the future as well. Next slide. So just to recall the central guiding principle of ACT, the focus of the Accelerating Clinical Trials Consortium is to accelerate, optimize, and facilitate the conduct, implementation, and results translation from high quality, high impact randomized controlled trials to improve health in Canada and around the world across a large number, I would say, of illness or disease states. Next slide. Starting first with contracts. So, I think it's very well recognized that contracts continue to be a bottleneck in conducting RCTs. ACT is moving forward to try to implement some shared agreement structures around contracts to try to help facilitate or make these processes a little bit easier. I do have an updated slide deck. I'm going to share that with Omo, and hopefully we can share that with the members, because there are actually some links to documents for those that are participating in certain institutions if you want to access some of these documents as well. So just in the updated deck, which I'm not sharing with you right now, it has some of those links. So just to highlight, these include data sample sharing agreements across 53 Canadian centers. And then they're in the process of finalizing a master clinical trial agreement for Tri-Council funded trials, with a goal of this being completed in June of 2025. Not sure how this will work, but if it does work very well, the goal is to sort of like streamline what is really a bottleneck, I think, that we all face when conducting clinical trials in Canada. Next slide. From a regulatory standpoint, a lot of this work did take place at the last meeting for those that were there, but ACT has kind of provided lots of feedback to Health Canada and recommended a lot of changes to the ICHE6, and then also subsequently contributed to some international efforts to sort of inform the WHO guidance for best practices for clinical trials. Again, there are some documents that sort of accompany this slide that in the updated deck we could send around, but I think through a series of workshops at the last sort of PAN consortium meeting, I think a lot of us provided a lot of feedback, which I think was quite valuable and hopefully informed some improvements to this regulatory process. Next slide. This was perhaps one of the largest achievements and really was, I would say, one of the biggest focuses of the first phase of ACT. Again, I think previously might have heard about how successful the UK was at doing clinical trials. And one of the strategies that they used is ensuring that they had research support, human resources at a number of community hospitals in and across the UK, so that when they needed to do trials, they were able to activate these quite quickly. So in the initial ACT grant, about $6 million was put away to activate 20 community portfolio hospitals, not academic centers that were already involved in trials, but community hospitals that weren't. And at each of these sites, a coordinator was embedded to try to help bring forward in a quicker fashion clinical trials, largely led by the networks that were part of ACT. So this was quite a sort of a heavy, time-intensive process to identify these sites. I'm fairly certain that some of the sites have been participating in and recruiting patients to CNTN, or sorry, I should say trials led by those that are involved in the CNTN, which has been quite a success for us. And recognizing how important it is to sustain this portfolio hospital group, Dr. Jennifer Sang, who I believe is based in Niagara, is now going to be leading a new ACT subcommittee focused largely on these portfolio hospitals. And I do believe in the next sort of iteration of ACT, there's going to be a focus on adding more portfolio community hospitals to further sort of enhance that ability to update clinical trials easily across Canada. Next slide. One of the other big stress points from terms of a bottleneck and also making things easier is, we can all appreciate that REBs can be sometimes quite onerous and difficult and don't necessarily always achieve what we really want them to. And so with that in mind, ACT put forward an RFA with a focus of creating a pan-Canadian single REB approval process. With a goal to sort of smooth out, the steps required in and around ethics and consenting, and of course the process around REB as well. So in February, there was a meeting of key leaders, and I believe patient representatives were included at that meeting too. May 2024 is where the RFA was announced. In August of last year was the application deadline, and there was an open competition with an objective process. Next slide. The selection committee is listed. I recognize all the names on here except for, I think, Levin. I haven't seen that name before, but all the other ones I recognize. No, I'm just kidding. Actually, I'd say this is quite exciting to know that, Adeera was part of this selection committee. Again, it just shows you how much involvement our nephrology committee, community, I should say, has had in a lot of the processes within ACT. So great to see that. And if you want to go to the next slide. And I think this is the successful candidate. So can review, there's a link there. And again, in the updated deck that I'll send through, I believe there's actually further information there as well. But ultimately, can review, I think, is finalizing this process, and the plan is to use sort of a digital platform for ethics approval. And, the key part is, sometimes as a network, it may be hard for us to specifically kind of navigate this from the network side of things. But to our investigators, this might be very helpful at the institutions that you're located where, you're submitting your REB proposals. So I think there's going to be some value in making sure these sort of documents about the single REB process and how to access it are there. I don't know if all institutions at this point are necessarily linked in yet, but something that will be, certainly valuable for the investigators on the ground. So like I said, I'll send that in the updated deck. Next slide. ACT has had a number of requests for applications across different areas. A lot of these really tied back to, what the government really wanted in terms of not just expanding clinical trials, but improving access to Canadian biotechnology companies as well. This is just a list of the 6 RFAs that have been completed at this point, but how much funding was allocated to each of them. And then next slide. This is just to sort of highlight some of the big RCTs that were funded by ACT. You can just see that they've had publication, of course, in some very high impact factor journals that are known to us. Next slide. In terms of knowledge mobilization, in February, the knowledge mobilization RFA was announced. I'm not sure about whether CNTN had an application in this, although we've had them in a number of other RFAs, as I'll highlight. I won't say much to this slide other than to say that there was some efforts, of course, in trying to ensure, the knowledge from these large trials that are being conducted are sort of properly disseminated out to the public. And I think we will hear about some of the successful awardees of this in early June. Next slide. And this is just a summary specific to CNTN. I provided that list of RFAs of how successful researchers from the CNTN have been in accessing these RFAs. And I think an important point here is that, if our network were not part of ACT, unless through another means, it would have been very hard for our investigators to be able to access these funds regarding these specific RFAs. So it does really highlight the importance of why we ultimately were in ACT and how it's kind of benefited us. I'm not going to go through all of these trials right now, but I think there may be further discussion later on, if not in this meeting, then in another one. But I really just wanted to highlight that we've been highly successful in getting additional research funding for some pretty big and important clinical trials. Next slide. And, part of that ability to get funding for RFAs also hinges a little bit on us being involved within ACT. And this was really a thank you from the operating committee and Aviva Blacker, really just to highlight that we have had representation across a number of ACT's committees, including, Research Network Committee, which is myself being a part of, and Clara, who's now in the co-chair position, patient engagement, scientific operations, and the scientific committee as well. We've also not only submitted a large number of RFA applications, but many of the members of CNTN have participated in reviews of RFAs as well, and the applications that are coming through. And we've had pretty regular attendance at all of the ACT meetings. I know there's going to be another one planned for the fall of this year. I don't know a lot of details yet, but hopefully we can maintain that attendance. Next slide. So this is not from me, but this is from really ACT itself. So, the view was that a lot had been achieved, and believe it or not, this is a 2-year time span, so a very, very short period of time. But there was sort of a view that, is 2 years and then another year left really enough to actually create sustainable change? Next slide. So with that, there is a plan for an ACT 2.0. Unfortunately, I was supposed to be part of the planning committee originally, but due to some unforeseen circumstances, I haven't been able to really participate. But there may be opportunities to do so. So certainly PJ and a lot of other members are going to be participating in a kind of a committee to start thinking about ACT 2.0 with the goal of seeking more funding to try to really solidify some of these larger system changes that are only really happening towards the end of this kind of first phase of funding. So that includes, success of the single RAB review, perhaps adding more portfolio hospitals, and then also ensuring that personnel are sustained at the existing portfolio hospitals with the hopes that there can be ongoing clinical trial enrollment, sort of ensure the implementation of that master clinical trial contract, and then to continue to help sort of build that bond between Canadian biotech and researchers through probably some additional RFAs in ACT 2.0. Next slide. There are also going to be some new initiatives. Some of those include, important steps around informing patients themselves, and I'm not privy to exactly what the structure of this is, but certainly some efforts there. This one I am, and there is going to be a request, certainly of CNTN, to come up with a contact list of all of the individual members within CNTN to try to, especially those that are sort of operating as a PI or a site PI, to try to really facilitate a process through which these individuals could be contacted for trials, both industry and non-industry trials that might be occurring at that site. So more to come on this. We do have a meeting that we're trying to get set up between CNTN and ACT, and I sort of suspect this ask is going to come through that meeting. And then to also look at certain populations that are largely excluded from trials to ensure there are strategies to perhaps include those patients and individuals to sort of enhance equitable access. Next slide. So this is kind of the ACT summary. I think we all know what ACT is trying to achieve. It was quite a big initiative. I would say they've been pretty successful in a lot of ways, but there's a lot of work to be done. And maybe through this ACT 2.0 with some sustained funding, there can be sort of further growth and further support. Next slide. And I'll pause there for any questions. I will emphasize, I am not part of the ACT operations committee. I have a role more focused on the network committee lead itself. So some of the questions around, where we are with certain updates with operations, I may not have information to that, but I can certainly do my best. And for those CNTN members that are part of some of the other committees, if you have anything else to add as questions come through, by all means, please do so.
All right, we're gonna applause. Thanks, Karthik. I hope you can hear that. So we have about
I can see your hands moving back and forth.
So we have about 10 minutes for questions and discussion. I want to talk a little bit about one of the committees that I'm involved on ACT. So it's the IDEA committee, which stands for Inclusivity, Diversity, Accessibility, and Equity. I think I mixed up those letters. So, and it's called lots of different things in different spaces, but the principles matter about access to trials so that everyone has a fair chance to participate no matter where you live in Canada, regardless of where your site is. So I'll just highlight some of the work that our committee has done. Shamala is a postdoc from Newfoundland who's currently in Montreal working with Dr. Louise Pillette, who heads the committee. We've completed a meta-epidemiologic study. So that's a study where we reviewed all clinical trials in Canada over the last 20 years registered on clinicaltrials.gov that have been only done in Canada and looked at lots of representation across different patient level factors. So things like sex and gender, things like they're called progress plus factors. So socioeconomic status, income, culture, religion, language, all these things that could potentially affect participation in clinical trials to highlight some of the gaps in different acute and chronic disease areas. So that was presented at the most recent ACT meeting, and it was presented at this, or it's going to be presented at the Society for Clinical Trials, which is in Vancouver. So exciting work from that space, using that work to answer questions in a qualitative manner, doing interviews with research teams, including investigators, research coordinators, patient partners about, why are these gaps? Why are they systematic? How do we fix them? What can we do to that? And that leads into solutions to barriers for participation, which I think is important. Lastly, we're also working on a scoping review of toolkits to improve EDI within clinical trials in Canada and internationally. And I think this is really important right now because if you look on the NIH website, everything is now gone. Things have been removed about diverse participation in clinical trials, how to optimize sex and gender representation, pregnancy, lactation factors, all those types of stuff. So there's an opportunity and gap for Canada to lead the world in this space. And I think this committee is really poised to make sure that not only in Canada, but internationally, we provide guidance to investigators and teams for these types of things. So that's just one of the updates from one of the other committees. I know CNTN members are planted in all types of committees. I think we're really proud of our patient engagement representation within ACT. I'll just shout out to Arlene, who I know has been really visible and vocal and present at a lot of the meetings, making sure that the patient voice has been heard. I think we all really respect that, and we're very happy to see, we're putting our foot forward about what we do within our own networks on a national setting. So I'll open it up to see if anyone else has any other questions or comments or discussion from participation within ACT or from the presentation. The one other thing I want to mention is it's so great to see these contracts and can review and stuff like that for Beat Calci, which I'm going to be talking about now, actually piloting using these new templates just to see. They're pretty long, but it's going to narrow down the back and forth that we've all seen across institutions, even though we work with each other all the time. Every new study, there's a new consideration, and I think the lawyers enjoy it, but they have bills to pay and work to do. But yeah, I think that's progress, and that's what we want to see. And I think within ACT 2.0, there's still lots more work to do. And thank you for providing the update and for your presentation, Karthik.
No problem. And thanks for highlighting the work of the IDEA committee. I did want to look up, certainly for the REB, the single REB, how much involvement there was, in particular from patients. And in the file that will be sent around, there is sort of a link to what CAN review is, who's involved in it, and I was happy to see that there was certainly patient involvement, caregiver involvement. I'm hoping that there was enough because I think informally, I had heard from patients since both CNTN and also outside of that just how important fixing that REB process is. I was also happy to see that there was involvement from certainly an Indigenous Research Ethics Advisory Group as well. I'm not sure who's part of CAN Review, but like in full, but there is a list of individuals that are there. And I think we have to keep pushing that message through. David, you talk about the States and what's going on there. I kind of fully agree with you that not only do we have the ability in Canada to sort of be leaders, but again, I'll say this repeatedly, I think our network, I feel is sort of light years ahead of a lot of other networks when it comes to engagement of Indigenous people and also of patients in general. So I think we are really working hard on that front, and we have a lot to share, hopefully in Act 2.0 as well.
Great, thanks so much, Karthik. I don't see any other questions or Kathy.
Hi, my name is Kathy Woods, and I'm the co-chair along with Arlene Desjarlis of IPERC for CANSOL. And I think one of the things that, I appreciate the work that you're doing on the scientific in terms of idea. And I think I would like to see in terms of First Nation, Métis, and Inuit involvement, in terms of patient partners, having a few more patient partners, but also taking the scientific and the idea stuff to the main part of the 2.0 of CNTN in terms of embedding that within your terms of reference or within whatever drives your organization together, and helping it move forward, because I think that's vitally important in terms of that, because one of the things about access to trials, in terms of there's so many barriers within the Indigenous community about being involved. And so, we at IPERC and that can help with that process and can keep moving that process forward. And we have a lot of access to different Indigenous advisory committees within Diabetes Action Canada. We're starting to work with lots of different organizations, Indigenous, FINISM, FNHA. So we've got a lot of different contacts and stuff out there to be able to help you accelerate that process.
Thank you, Kathy. And I just want to share a little bit just further that. So Kathy and I are working on a clinical trial in First Nations communities in Manitoba to screen for kidney disease. It's called Virtual Kidney Check and Follow-Up. Indigenous-led research question team with community engagement. What we found in our meta-epidemiologic study is Indigenous patients are not represented in clinical trials in Canada. And there are multiple reasons for that. And there has been qualitative research diving into that questions. There are systematic barriers for participation, retention. I think one of the big thing is, are we asking the right questions? And are we with the right teams, with the right people? So I think there's indigeneity is sometimes it's viewed separately within EDI. And we're really lucky to have Wayne Clark from the University of Alberta with an ACT that's providing an Indigenous lens on everything that we do, and it echoes a lot of the comments that you talk about, Kathy. And I think here at CNTN, we have a privilege to have the patient voice at the front, to have Indigenous patient partners and IPERC. And I've had the privilege to work within the Manitoba, Finism and Kim and other people with Barry Lavallee and Paul Commenda, even though I'm not in Manitoba anymore. But I think a lot of lessons learned that we can share throughout the network to build to make sure that Indigenous patients have access to clinical trials, not only in nephrology, but within acute and chronic diseases in Canada. Thanks. Okay, any other questions? All right, thanks so much, Karthik. I'll take over from here. So I'm going to be talking. Thanks. So I'm going to be talking about RFA number 6 or 5. I think we've lost track of what we're on. So this is called Beat Calci. So Better Evidence in Translation Calciphylaxis. It's an adaptive platform trial. And so the team from the University of Sydney in Australia, so Meg Jardine and Rathika Krishnami, have been leading this trial globally for the last couple of years and have been on a mission to involve other sites from across Canada. I applied to CHR 3 times over the last 2+ years to get funding for them. It got triaged every single time because 2 things. One is they had problems with the outcome, which, it is what it is. But the second one was you don't have Canadian pilot feasibility data. This is a multi-center international trial that's already recruited 30 to 40 patients. We're not going to get feasibility data. So I gave up at CHR, but once I saw within ACT this opportunity, submitted it. I worked with a large group of people. A lot of people in this room have been involved so far too. And we were successful, and I'm excited to share information about this trial. So this is one of multiple clinical trials within a breadth of acute and chronic diseases that were funded. It's the only nephrology one, but if you look, there's some heart failure trials, some vascular disease trials, a lot of stuff related to stroke. It's from across the world, bringing it to Canada to give opportunity to participate in clinical trials in the UK. There's ones from Germany, from the United States. So this is one, otherwise patients wouldn't have access to this clinical trial if it wasn't for ACT, because of the challenges in the current funding framework within Canada. Okay, I've presented this slide before in Alberta, and I got some negative feedback from people about why did you put that picture up in the room when so many people are there. But this is what we see clinically. Calciphylaxis is an awful disease with awful outcomes, and there are no clinical trials that have successfully been done to inform the care of this patient population. So the other term for calciphylaxis is calcific uremic arteriopathy. It's calcification of the small vessels in the skin and the fat. It's really rare. You might see one or two every year. There's pockets of variability. I saw a lot more in Hamilton and Winnipeg than I have seen in Edmonton, but there is variability. We don't know why. It's very serious. Mortality at 6 months, so death is 50%, and there's lots of painful ulcers, poor quality of life, prolonged hospitalizations. And there's often delays in diagnosis that can make management challenging. So this is a very severe disease that we don't have any evidence-based treatments for. So there's a big gap that we need to address. So the pathogenesis, there's lots of different factors. It's basically an imbalance of procalcification versus anti-calcification of these different factors. Things like calcium, phosphate, parathyroid hormone, vitamin D, FGF23. These are all hormones that crosstalk with the bones and the kidneys and the parathyroid glands. And there's some other markers that are elevated that are potential targets. But the bottom line is this imbalance causes the calcification. There's lots of different risk factors that are seen in observational studies and registries. We don't have a registry in Canada, but there's one in Germany, there's one in Australia that have found, why do some people get it? What are the outcomes? But there's really no clinical trials in this area. So there's really this unmet need. There's been no large RCTs. There are no proven treatments. We don't even have a standardized method to assess calciphylaxis outcomes, which is one of the criticisms of the review that I mentioned before. So embedded within the trial, we're validating this ordinal scale and doing some work with photography and AI and standardized measurement of wounds. And there's a lot of different clinical outcomes that are relevant to patients, caregivers, and clinicians. And there's this urgent need to expand that evidence base. Clinically, I can tell you what my practice is I don't know if it's safe and I don't know if it works, but it's such a bad disease that I want to give everything to everyone. And they're costly, there's side effects, they might cause harm, but we're so desperate. And that's supported by survey work we've done in Canada. So Beat Calci, it's an investigator-initiated and international, so it's in Australia, New Zealand, Denmark, and now Canada, which potential expansion to other countries. It's a platform randomized control trial, which I'll talk a little bit about, and has multiple domains and multiple interventions. And really the goal is to establish high-quality evidence for the treatment of calciphylaxis in the setting of kidney failure. Won't go through all the secondary objectives because they're focused on different outcomes, but we want to look at the ulcer size, pain, quality of life, medication use, hospitalizations, things that are relevant to different types of key informants in this area. So it's a multi-domain platform trial. So this means that there's multiple domains or therapeutic areas. There's 2 that are active that are coming to Canada. There's potential for more, but we're looking at pharmacologic therapy, so medications first, and these are pills or intravenous medications given during dialysis. So focusing on that. The other domain is dialyzer membranes. So looking at medium cutoff dialyzers versus usual care dialyzers. We Someone can be in one domain, they can be in multiple domains. There's flexibility, there's comparison to placebo or active, and it's an efficient way to answer the question in multiple time. And there's just that flexibility. So the potential future domains down the line are things related to wound care or hyperbaric oxygen. If there's something new that comes out, that can always be added as well. So for the eligibility for the trial, so there's a core protocol that has the key eligibility that apply everywhere, and it's basically an adult above the age of 18. You have to have kidney failure and be on dialysis with new calciphylaxis for less than 10 weeks, and you have to be eligible for one of the treatments. We've done survey work. There's definitely equipoise in this area that people want to enroll their patients in these clinical trials. The overall exclusion criteria for the platform, there's none. They're all specific to the domain-specific appendices. So in the dialyzer membrane one, it's basically, are the clinician and patient willing to provide consent and participate, and that they're not using hemodiafiltration, which is not widely practiced across Canada outside of Quebec. So this is what the membrane design looks like for the platform. So it is a one-to-one randomization of the medium cutoff dialyzer. So it's called Theranova 300 or 400, and it dialyzes middle molecules that are those procalcific molecules that we're thinking about compared to usual care. There's follow-up at 4 weeks, 12 weeks, and then 26 weeks. If at 12 weeks that there is no response, you can switch to open label. So it's really important to have flexibility and adaptation embedded in this clinical trial because patients and clinicians are really worried about when things don't get worse or things get worse. In the pharmacotherapy domain, similarly, have to have consent and support from the patient and clinician. And there's things that are specific to the interventions that you may be able to not tolerate or other exclusion criteria, but I will go over them in detail. So the interventions for this one, it's placebo controlled. So there's IV sodium thiosulfate given 3 times weekly on dialysis. It can cause wicked nausea or acidosis. It's very expensive. And the most recent meta-analysis of observational studies actually shows that it makes absolutely no difference. So that's one of them. Also, vitamin K and magnesium citrate are other potential interventions involved in the pathophysiology of the disease. So it's a placebo-controlled trial. Everyone gets at least one active medication at the start. There's the potential for rescue medications at 4 weeks and 12 weeks that you can add active therapy on top of that, depending on standardized outcomes. This is just a follow-up that shows after the baseline at 2, 4, 6, 8, 10, 12 weeks, there's always an assessment of pain and co-interventions for analgesic, but the main outcomes are done at 4 weeks and 12 weeks and 26 weeks related to wound size. So we're looking at this, it's called Beat Calci Wound Assessment Scale. It's 8 points. It's a categorical scale ranging from 1 to 8. So 1 being the best outcome, so that there's complete healing of the ulcer, to 8 being the worst outcome, which is death. So it's a categorical scale. It has great statistical properties. These are meaningful to patients and clinicians, and it drops your sample size. And we want to validate this outcome as part of the trial. This is used in a lot of other diseases like stroke and stuff like that related to outcomes. It has really complex Bayesian statistics that I don't fully understand, but really people smarter than me from Australia and the Berry Group from Texas have done. But ultimately, there's 300 patients in each domain. It has an 80% power to detect an odds ratio of 0.5. They've done these complex simulations, and there's interim analysis every 48 patients. And they almost are approaching their first interim analysis from Australia and New Zealand. There's been great recruitment so far. So this is the global snapshot. It's a little bit outdated. We actually had a call last week that it was provided updated, but it's in Australia, New Zealand, Denmark, now Canada. We're 10 sites so far. We're getting started with the dialyzer one right now because it's easier to get started. There's been some barriers from Health Canada getting approval across the different domains. But thank you for the people in this room that are participating. And it's really a national effort, but hoping to expand it as well with funding from Australia and other countries getting funding. Denmark actually got funding from Novo Nordisk. Not all countries have ACT, so we're really lucky, but it's been a challenge so far. So I just want to thank the group in Australia and New Zealand to bring Canada to the table, the ACT, the CNTN, which provided letters of support for multiple submissions across granting agencies and my different institutions. I think this is a really exciting type of trial design, which we kind of started to see in COVID, of these adaptive platform trials that test multiple interventions. They're efficient, they're patient-centered. They answer multiple questions. They're adaptive in the sense that you can add or subtract standard of care or something that you actually become standard of care as part of the trial. You can do multiple domains. You can re-randomize people if they're not doing well. You can actually shift your ratio of randomization. If you start seeing people doing well in one arm, you can start funneling more of them. And this is all possible in the Bayesian framework, which is really exciting. So I'm going to learn lots more skills in this area and be supported by the team there. But this trial is not possible with the network of investigators across Canada that will be recruiting their patients. So thank you. We'll open it up for questions. Time for one or two questions. Cal.
A very simple question. Thank you. Yeah, simple question. How do you go about blinding for the dialysis membrane? If they're different sizes, colors, things like that, just out of curiosity.
Yeah, so it's not feasible to blind. You know, at a clinician level, you're going to be ordering it. At a patient level, they can see it. So we're not going to cover it. It's just one of the things that we've accepted. From the pharmacotherapy domain, we've invested the time and energy to make matching placebos. But the dialysis membrane doesn't have blinding.
Hey, thanks for the talk. I just had one question. I'm more curious about your opinion because, as you mentioned, a lot of this is already set before you. The power is 80% for 50% equivalent relative risk reduction. So, like, do you feel like, or have you guys talked about missing moderate size effects, like 40%, and being underpowered for that, and for something we don't have treatments for?
So that's always the threat, right? Of a balance of what's feasible based on numbers and your budget. There is some interesting support for this too. We're lucky that there's not the issue of multiplicity, so multiple hypothesis testing across all those interventions domains. But you got to pick something and defend it. I think it's that odds ratio in the setting of that it's an ordinal outcome for a change in one of those scales. But I would love 90% power, and I would love for the odds ratio to be 0.7 or 0.8. That's something you think is there, but that just inflates everything insanely. And just from a funding perspective, even trying to get this for feasibility, it was the barrier. So I think we can always revisit what it looks like and do interim sample size adjustment in a Bayesian framework, which is one of the adaptive flexible parts of it. Great question. Okay. That's all for me. So we're going to move on to the next presentation. So I'll get hands back up here to introduce Dr. Adeera Levin. And does my clicker actually work? Are you helping me? Sorry? Okay, I thought so. Or I just had confidence going through. Right, Hans?
Well, good work, David. Very interesting. Actually, even for a layman like me, I'm quite excited about it.
You taught me how to do that, though, Hans.
It's a great pleasure for me to be able to introduce some very smart people, and Adeera is certainly one of them. She is a lot of things, but she says, "Don't introduce me because everybody knows who I am." So, following her wishes, I will not introduce the eminent Adeera Levin, who is going to speak to you very briefly on Easi-Kidney trial.
Thank you. Now, this does or doesn't work. So I'm going to, some of you have heard this before, but I think it's really worthwhile going back again to give you a little concept about the rationale and design of Easi-Kidney. And many of you participated in EMPA-Kidney. Can you raise your hands if you did EMPA-Kidney in this room? No, a few of you. Well, our study coordinators. Okay, so this is, so does this work? Next. It doesn't work. Okay, next. So this is an opportunity to actually better understand the aldosterone pathway. And this is one of the pathways that's important in both scarring the kidney as well as high blood pressure, as well as scarring the heart. And so it's important. Can you just keep clicking and make this all come up? So we know these drugs called ACE inhibitors and ARBs, and we also know about spironolactone and finerenone. So this new drug actually blocks aldosterone higher up the same way as an ACE inhibitor, ramipril, blocks instead of an ARB blocking. So it's another way to block the aldosterone pathway, which is important in what I said, in scarring the kidney and the heart. Next. In a number of studies, Fine-Heart, Fidelio, Figaro, and Fine Arts heart failure, we showed that, or people showed that blocking this aldosterone with a mineralocorticoid receptor antagonist actually leads to good outcomes from heart disease, hospitalization, and in delaying progression of kidney disease. So we know this is an important pathway. Next slide. These are just the odds ratios that David was talking about that he wishes we had in other studies, but these are very important changes in outcomes for patients. Next slide. So in these large studies that have been looking at the mineralocorticoid receptor antagonists, a number of people were not included. Most of these studies are with people with diabetes. So Easi-Kidney is trying to look at people without diabetes. And a number of the studies looked at people without with lower levels of protein in their urine, people who were not able to take a RAS inhibitor, people who had high potassiums were excluded, so you had a potassium lower than 4.8. And so also people didn't look at, in the other studies, at those with lower GFRs and those who are on SGLT2 inhibitors. So this is, we were talking before about EDI, like there's a group of people who've never had this mechanism or these drugs studied in them. Next. We know the SGLT2 inhibitor story, and those are the Flozin drugs, and we know that they're amazingly powerful at reducing outcomes. And this is just a summary of that, but that's a 20 to 30% risk reduction in all outcomes, kidney, heart, hospitalization. And so they're an exciting agent. Next slide. They also reduce hyperkalemia, which is important because that's one of the things that limits us using these aldosterone blockers. And so in Easi-Kidney, the rationale is that we need these very large trials, so quite different than the adaptive trial that David just talked to you about, but really large trials to try and define and assess how safe it is to block this aldosterone pathway in this location. And there's these new medications that are ASIs, because it's easier to say, but they aldosterone synthase inhibitors. And we want to also make sure that we study non-diabetics, because it's fine to keep having all these things for diabetic people, but we don't have them for non-diabetics. And so we wanted to also look at whether or not we could combine the ASIs with what we know really works well, the SGLT2 inhibitors, to protect people, but also we know that they work to reduce potassium, which is one of the rate-limiting steps for these other drugs. And we can also, many of us in the nephrology world don't get so excited about potassiums of 5 and 5.1. And so all the other studies were incredibly conservative in the entry criteria for potassium. This is also another attempt to look at studying a higher potassium group, which is a lot of people with kidney disease. Next. So this is what it looks like. It's people at risk of kidney disease progression with and without type 2 diabetes, with 4,800 people in the diabetic group and 6,200. So this is the largest ever kidney trial with 11,000 people trying to get randomized. Everybody gets a SGLT2 inhibitor. So in those provinces or places where SGLT2 inhibitors are harder to get, or there are limitations, this would be a huge incentive because you'd get put on that for sure. And then you get randomized to what's now called, I won't be able to say it, versus placebo, or the ASI versus placebo. And so that's nice. And then what we're looking for is cardiovascular death, hospitalization for heart failure, or kidney disease progression looked at in the usual ways: kidney failure requiring dialysis or transplant, or a sustained decline of 40%. So these are conventional things that we now look at that are important to patients and to healthcare professionals, as well as to healthcare systems. It's event driven, so we need 1,000 primary events in every stratum, and we'll have a 90% power to detect an 18% risk reduction. And why that's lower is because now you're already giving people an SGLT2 inhibitor, which we know reduces risk substantially, and we can't get greedy and look for another 30%. So what's nice is you don't have to be on an ACE inhibitor to get into this study, which is quite nice because all the other studies say that you need to be. Next slide. So the recruitment targets are 12,000 to end up with 11,000. We've screened, as of April 25th, 1,200 people and randomized 242, so it's launching. It is truly an international study. Canada is supposed to put in 600 patients. We're hoping to do more than 600 patients, but let's try and get to the 600 quickly. Next. We have multiple sites. They're not all, quote, academic centers, which is great, much to what David was saying, but the ACT trial is trying to enroll many others. So just keep flipping. So it's British Columbia, Alberta, Ontario, multiple sites, Quebec, multiple sites, Nova Scotia. So that's pretty great, right? It's like almost pan-Canadian, except for Newfoundland, and there are various reasons for that. Next slide. So the first participant screened in Canada was in Vancouver on March 18th. And the first one was randomized on May 13th, which is coming. And what's really nice, not yet, going to be. So our current Canadian numbers are, to our little contract drama that we talked about, we've only had 22 of the 23 contracts, but they are hugely problematic. And so I'm looking forward to ACT fixing that problem for us. 2 sites have been activated, Vancouver and Edmonton. 146 patients have been screened, pre-screened. So this also predicated on pre-screening your clinical rosters, if you will, to actually see who's most eligible. That makes it much more efficient. We've actually screened 4 participants, but we have, like, as I said, there's 2 people in queue to get randomized. So that's not bad, but now we have to pick up the pace. Next slide. So this is just like a summary of the study, and then I'm happy to answer any questions, but this is really an opportunity for us to participate in a huge international trial, but really also establish a bit of an infrastructure for ongoing clinical trials. And that's what I was talking to Io and others about. Like, if we can get this up and running in that many sites, then how do you sustain our clinical research teams in those sites so that we're prepared to do other studies, including adaptive platform trials and other things that are coming? And maybe if you can be in Beat Calci and in this, like, that's sort of another way to just keep your teams together so that you don't have to restart them each time there's another study. So shout out to our local coordinators and Zainab for all of her patience in the contract negotiations with every single thing, and Amanda for all of the work that she's done training across Canada, because getting everybody up so you can train it. The other important thing is the Oxford Clinical Trials Group is very committed to making trials simple. So the burden of visits here is quite low after the initial run-in phase, which is really great. So the idea is you put them into the trial, and then it's the usual clinical care, and then you just keep going. Yeah. Thanks. Yeah, so it's very exciting. We're trying to be very inclusive. It is a new agent drug, but it's been tested and shown to be of benefit in smaller studies of 200 people. And it's another part of the pathway. And we're thinking there are a number of people who aren't very good on ACE inhibitors who would be good for this drug. So questions or thoughts? Yep.
Any idea what the blood pressure response on that is?
It's less than the other drug, the other ASI, but it's about 2 to 4 millimeters of mercury, right? Yeah. So stopping scarring of hearts and kidneys is a pretty big, pretty exciting time. We have lots of different agents to do that now. One of the things that we're also working on is this is in adults, but I'm very conscious of the fact that children should be, or at least adolescents should be able to be involved, although it is a hormone blocker. So it blocks a hormone that might be important. So it may have different things. But in talking to Louise Ono, one of the things that we're trying to do is get the age of trials to go down to 15 instead of 18, because the 15-year-olds are sufficiently similar to an 18-year-old. And FDA actually allows you to make the case of sufficiently similar to enroll people at different ages. So it's important to think about that. Okay. All right. So whoever is at the sites, and some of you I see, like, just pre-screen makes a huge difference, and then like enroll quickly. Great. Thanks.
Right, so we have about 5 minutes for questions or discussion. My site is active and we've pre-screened, but I'll just mention that our REB doesn't let us pre-screen anymore, but we got the advantage of a reciprocal exchange with the University of British Columbia so that we were allowed to pre-screen for this. And I think we've benefited from that relationship between another institution. So with CAN Review coming out and with AC2 2.0, that idea of access informed to all patients to hear about things, I think pre-screening is part of the dialogue that's happening nationally. So thanks to Adeera for helping us out with that with our site. So I'll just mention, so Adeera talked about the other drug. So I'm doing the Baxdrostat trial, which is it's by AstraZeneca. It's an industry trial. It does not heavily involve investigators in the design and conduct. It's a very different trial. It is not as pragmatic. It has more stringent eligibility criteria, but it's great to see lots of activity in this space in Canada. There are different types of patients, different risk profiles, same intervention, but it's a very much more potent antihypertensive. So Dr. Prasad asked that question. Baxdrostat drops it off a lot more. But it's great to see industry engagement within Canada. And as Adeera mentioned, we want to see the pipeline and these relationships with Boehringer Ingelheim and Oxford and AstraZeneca. And, we've all seen the exhibition people out there, that we want them to keep coming to Canada because our patients do well in the trials. We recruit those numbers that we want and we add to the science. So I think that's really important. So I'll ask if anyone else has any questions or comments.
Make one comment. One of the things that's really interesting is when people say, well, I'm going to get randomized, like that means I won't get something. No, you're going to get all your usual care, and you're going to have two sets of eyes looking after you, your clinical eyes and the research eyes. And I think that actually is really reassuring to people, that you actually have, it's actually better. And there's lots of data that suggests that many of us know that actually being in a clinical trial, you get better care, even if you're in the placebo arm, than not. And I think that's a really important thing to say to people. That's not coercion, that's just telling them the facts. And I think it's really important that we as a community understand that as well.
Great. Right. So if no other questions, we'll move on to the next presentation. So I have the pleasure of introducing Dr. Vikas Sridhar. He's a transplant nephrologist and clinical assistant professor within the Division of Nephrology at University of British Columbia. After completing nephrology and transplant training at the University of Toronto, he pursued a PhD in mechanistic and clinical trials in cardiorenal disease under the mentorship of Dr. David Cherney. He has multiple research awards and over 50 publications. His research interests include the use of cardio-kidney metabolic therapies in transplantation and the interface of cardiac and kidney disease. Please join me in welcoming Dr. Vikas in presenting the SugarnSALT trial.
Thank you. And I'd like to thank the organizers for the opportunity to present SugarnSALT. Next slide, please. These are my disclosures in the next slide. So topics for discussion today, I'd like to just quickly summarize current strategies for kidney protection in people with type 2 diabetes. And then I'd like to focus on SGLT inhibitors, sodium glucose co-transport inhibition, focusing on some of the evidence in people with type 1 diabetes. And then I'll wrap up on our efforts to actually study these agents to protect kidney function and prevent loss in people with type 1 diabetes. So next slide, please. And the next one. So many of us may be familiar with this pyramid. This is from the KDIGO guidelines on the management of people with chronic kidney disease and type 2 diabetes. There is a new figure. This will be updated, but I just wanted to highlight these 4 agents over here: RAS blockade, SGLT2 inhibitors, MRAs, and GLP-1 RAs, just to show that we firmly are in the era of 4 pillars for the management of diabetic kidney disease. Next slide. However, we do not currently have evidence for the use of those agents in people with type 1 diabetes, as they were all almost universally excluded from all of the large trials. So there's a persisting and, in most of our opinions, serious problem of diabetic kidney disease in people with type 1 diabetes. It's a scourge. It occurs quite frequently. It is associated with morbidity and mortality. And when these patients do develop diabetic kidney disease, many of them do progress to end-stage kidney disease. Next slide, please. So all is not lost. There are some therapies that we currently utilize. There is intensive glycemic control, as demonstrated by the landmark DCCT EDIC trial, as well as evidence for RAS inhibition as well, which is what we currently use in these patients. However, despite this, there remains quite significant residual risk of progression of diabetic kidney disease to end-stage kidney disease. Next slide, please. So I'd like to focus on sodium glucose co-transport inhibition and the evidence for their use currently in type 1 diabetes. Next slide. So almost all of us in this room are familiar with the overwhelming evidence of protection with these flozins. This is summary data from the SMART-C meta-analysis, which includes all of the trials, almost all of the SGLT trials that have been completed to date in people with CKD, heart failure, and atherosclerotic cardiovascular disease. This particular panel focuses on the evidence for slowing down kidney disease progression. And there's about a 40% relative risk reduction, and that is agnostic to whether or not you have type 2 diabetes. In the next slide. This shows the same data for prevention of heart failure outcomes. It's slightly less. It's about a 25% relative risk reduction. Again, irrespective of whether or not one has type 2 diabetes. And the next slide, please. This one focuses on MACE. It's about a 10% reduction compared to placebo. And once again, it doesn't really matter if you have type 2 diabetes or not. And in all of these studies that were included in these analyses, there were very few patients with type 1 diabetes. The next slide. So we have been interested in the use of these agents in people with type 1 diabetes. There have actually been a number of studies with agents like dapagliflozin, empagliflozin, and sotagliflozin in people with type 1 diabetes. Most of these were smaller, so not 11,000, but maybe 100, 200. And most of these were low-risk patient populations focused on glycemic outcomes. We were interested in actually applying some of these risk prediction equations at baseline. We would calculate these scores, and then we would calculate it at the end of the treatment period. And we did this with dapagliflozin, which is this picture over here, and empagliflozin as well. And we were able to demonstrate that there are reductions in the predicted risk of cardiovascular outcomes and kidney outcomes at the end of follow-up period. The reductions in absolute risk are relatively modest because these are low-risk populations, but there certainly is some interesting suggestions that these agents are similarly protective in people with type 1 diabetes as well. And the next slide. However, we do not currently use these agents in people with type 1 diabetes, and it's mainly this upper panel over here, which is this increased risk of diabetic ketoacidosis with an odds ratio of roughly 3 to 4, depending on which study one looks at. Next slide. And the next one, please. So we specifically asked the questions as to, are there any differences between using these agents in people with chronic kidney disease versus not chronic kidney disease? was relatively loosely defined. We were working with the population that we had. This is based on the in tandem studies. This is something that Ayo and I worked on together. And we defined CKD as simply a GFR of less than 60 or the presence of microalbuminuria. And these are very small numbers, but we don't necessarily appreciate a difference in the risk of DKA between those with CKD and those without. And indeed, physiologically, there may even be an argument that people with CKD may have a lower risk of diabetic ketoacidosis compared to the general population. Next slide, please. So to summarize what we currently know about SGLT inhibition in type 1 diabetes, we know that there are A1C reductions that are relatively modest. There are blood pressure reductions of about 3 to 4 millimeters of mercury. Body weight reductions are also observed. There are reductions in the need for insulin. And there are reductions in renal blood flow, so reductions in renal hyperfiltration, which manifests as this acute decline in GFR and arises in the hematocrit. There are potentially some reductions in hypoglycemia, and there is an increase in ketogenesis, which is very much part of how this drug works. Next, please. So what we do not know is anything about cardiovascular and kidney protection in those with type 1 diabetes and diabetic kidney disease, as once again, they were excluded from all of those trials that I summarized in the SMART-C meta-analysis. Next, please. So then we'll transition to how Canada and among other countries are studying cardio-kidney protection with SGLT inhibition in people with type 1 diabetes. The next slide, please. So we actually feel there's a very strong rationale to study the use of these agents to prevent DKD progression. In the general population, when you do the risk-benefit analysis, you have these relatively modest reductions in A1C of about 0.4%, versus, as I summarized earlier, about a threefold increase in the risk of DKA. But when you go look at the panel on the right, which is looking at people with type 1 diabetes, but now moderate to severe DKD, now the top part of your equation, the risk, the benefit part of the equation is avoiding progression to kidney failure, which I think is a huge deal, versus a threefold increase in the risk of DKA, and time will tell potentially that's even a lower risk of DKA risk in these people. So we feel that there's a much stronger rationale to study it, not for glycemia, but rather to actually prevent kidney disease progression, and along with that cardiovascular risk as well. The next slide, please. And the next one. So this is a little bit about the birth of SugarnSALT. It started off with support from the organization formerly known as JDRF, Breakthrough T1D, which was very interested in supporting a pivotal trial to actually gain FDA indication for kidney protection. And the next panel, please. And then there was also support from CIHR and specifically the Kidney Foundation of Canada, under the leadership of David Cherney and many people in this room. We were successful in obtaining funding for a pilot study. And the next, please. And then we reached out to Lexicon, who was able to provide sotagliflozin and matching placebo. And next, please. And then that led to the creation of the SugarnSALT trial. And then finally, on the right, Dexcom gave us CGM devices as well for all participants. So the next slide. So this has led to the SugarnSALT trial with the aim of evaluating the effectiveness and safety of sotagliflozin and slowing down kidney function decline. This is not an 11,000 patient study; it's 150. But they will be after run-in period randomized to sotagliflozin 200 milligrams versus placebo for a period of 3 years. And then after that, there's a 2-month washout period. And throughout this period of time, we will be measuring eGFRs based on creatinine and cystatin. And there are two important aims of here. Aim one is to determine the effectiveness in slowing eGFR decline in these in patients with type 1 diabetes and advanced diabetic kidney disease. Equally important is aim two, which is to institute and subsequently assess the feasibility of a DKA mitigation protocol to actually ensure that, if we do have evidence of clinical protection, it is done so in a manner that is safe and that can subsequently be replicated by clinicians who actually take care of these patients. Next slide, please. And the next. So key inclusion criteria, they need to have type 1 diabetes on stable RAS. We're looking for an eGFR of 20 to 60 mils per minute and at least 200 milligrams per gram, so 20 milligrams per millimole in our language. The primary outcome, which the study was powered around, was actually the Siri's talking to me for some reason. Okay. Is the EGFR creatinine cystatin C after that 8-week drug washout, adjusted for baseline EGFR and baseline ACR. It was actually powered around a 6 mil per minute difference between treatment and placebo groups. There are several secondary outcomes as well, including the traditional kidney composite, albuminuria, EGFR slope, as well as cardiovascular outcomes as well. Next, please. So I did mention the risk mitigation protocol, which is a very important part of the study. There's a lot of, we're excluding people at high risk. There's a lot of education before the study, during the run-in, and over the course of the study. We have an expert panel that are going to be managing changes and limiting when possible changes in insulin. There is quite frequent ketone testing that is incorporated routinely as part of the study, as well as on indications such as with illness, sustained hyperglycemia, or any symptoms. And then when a persistent elevation in point of care ketone testing is noted, we have this STITCH protocol, which is to stop the SGLT2 inhibitor, administer insulin, increase carbohydrate intake and some hydration as well, after which we are able to reassess this. And if necessary, medication will be withheld as well. And the next slide. And there actually is evidence for such DKA mitigation. This was published in 2020 in Diabetes Care, that the implementation of such a protocol is actually quite successful in actually reducing that risk of DKA when looking at a pre versus post analysis. Next slide, please. So this is the study timeline. Man can propose, but contracts get in the way, so we're probably about a year delayed. And the next slide is where we are right now. So this is as of April 28th. We have about 54 that have consented, 20 that have entered the randomization period, sorry, the run-in period, and 10 that are actually been randomized. And every day more and more people are actually, more and more sites are coming up, and we actually just got the email from our DSMB from the first meeting that they were quite happy with the progress that's under the leadership of Dr. Gerstein, and they congratulated us on the current progress of the study. And with that, I will wrap up. Actually, maybe the next slide, which shows all of the sites and all of the investigators. I'd like to thank all of our coordinators, all of the sites, and of course, all of our patients. And we are excited to wrap up the study in a timely manner and actually generate some evidence for the use of these agents in these otherwise high-risk patient population.
We're a little ahead of schedule, so we have 10 minutes or so for questions or discussion, so we can open it up.
How many patients have you enrolled so far?
Maybe we can go to the previous slide. It's a bit higher now. I think since this figure was out, we have maybe 3 or 4. So it's about 14 that have been randomized so far, a target of 150. But there's a lag. So as you can see, we have 54 consented. So that number should start to become asymptotic soon, hopefully. Yeah.
My question's about, I saw CKD epi creatinine cystatin C as a secondary outcome. Why did you think that it was necessary in this patient population?
Yeah. So the primary outcome was actually EGFR based on creatinine and cystatin C. So it's actually the primary outcome as well. And that's just based on our power calculations and yeah.
So I'm gonna go out on a limb and say that SGLT2 inhibitors should be in the water, and proteinuria, GFR slope, kidney failure will go down. So that risk of DKA, I think, like absolute versus relative, right? So I guess one of the questions I have is, have you guys, for patient partner engagement work and clinicians, had discussions or surveys about trade-offs between the two from a safety perspective, and what type of
Yeah.
signal are you getting to look at a net clinical benefit?
Yeah, so we this was led by Dr. Bruce Perkins out of Mount Sinai, and we did so we had patient partners as part of the CIHR KFOC application, but then even prior to that, that application is based on some of the work that Bruce had done, where he actually interviewed these people with type 1 diabetes and actually did a sort of risk reward survey. And what we found is that these patients who actually, first of all, they were highly self advocating, and they were very much willing to actually accept this risk of DKA for kidney benefit, especially with the backup of with the backup of these DKA mitigation protocols. Aim 2 of the Sugar and Salt CIHR application is to actually study this it's a quantitative qualitative analysis being led out of Calgary by David Campbell, who's actually performing a discrete choice experiment involving patients as well as physicians to actually explore the risk-reward trade-off a bit more in a quantitative manner. Yeah, so that some of that work will be coming out, and he actually emailed us this morning. We've completed the first part and actually initial surveying to design this discrete choice experiment. So we will get a lot more on that in that space.
Yeah, thanks so much. It's super interesting, important study. Along kind of the same lines as David, I think the absolute risk of DKA is very low. Can we get better at predicting who those individuals are that may be at highest risk of DKA? I know you have some kind of basic exclusion criteria, but could this be an opportunity to develop risk prediction models and kind of build that into the study design as well?
Yeah, and certainly this will be a rich source of data in that space. I think I strongly do believe that the risk of DKAs is even lower in this particular patient population compared to your general population, simply because of that reduced glucosuria and reduced ketogenesis. So yes, definitely an opportunity. There do exist some of these risk prediction models right now for DKA. I don't think it necessarily applies too well to these patient populations that are a bit more advanced. But yeah, I mean, certainly there's going to be a lot more. So assuming this ends up being a positive study, there's going to be a lot more interest in perhaps using these agents early on. And certainly that's when some of these questions will come into play, because now you're not dealing with people with more advanced CKD, but rather people potentially earlier in their disease course where this is a more pertinent issue.
You have time for one or two last questions? Just want to make sure online there's nobody. Not yet. Okay. Don't be shy. Any other questions or comments? I got one last one. Sure. I'm gonna monopolize my role here.
Keep the mic.
Can you hear? Okay. So can you tell me a little bit about that run-in period and how you're selecting for like an adherent patient population, or what are the study procedures that are doing to enhance?
Yeah, so it's more about optimizing glycemic control, ensuring that they're on RAS blockade, and a lot of education is occurring during that 6-week period as well. There's also ketone monitoring that is going on during that particular period as well, looking at compliance and ability to actually do the testing on a bi-weekly basis and getting a sense of what these ketone levels that there are as well. So there's a lot of safety that's built into that run-in period, in addition to education, in addition to getting them on baseline glycemic control and RAS inhibition.
Great, thanks so much.
Yeah.
Thanks.
I am pleased at the moment to introduce Dr. Thomas Mavrakanas, a nephrologist, clinical scientist, and associate professor at McGill University. His research has been focused on the causes and treatments of cardiovascular disease in patients with chronic kidney disease. You might have heard of that. And end-stage renal disease, that one too. He has authored over 63 articles published in peer-reviewed journals and has received several peer-reviewed grants and awards. Please join me in welcoming Dr. Thomas in presenting the CARE-MRI trial. There's your clicker.
Excellent. Thank you very much. So thank you very much for the kind introduction, and thank you all for the invitation. So I will briefly present you today the CARE-MRI trial, and would like your feedback and input on some issues we have with the study. So next slide, please. So we all know, and it was already said before, that SGLT2 inhibitors improve cardiovascular and kidney outcomes in patients with preserved kidney function and in those with CKD. And most trials had stopped at the GFR of 30, but now we have several studies that actually include patients with a GFR down to 20. And in a recent meta-analysis, we showed that the cardiovascular benefit is probably preserved in this population too. Next slide. However, what is not known yet is the potential efficacy of these medications to prevent cardiovascular events in CKD stage 5, including patients on dialysis. So before designing CARE-MRI, we did a pharmacokinetic study with canagliflozin in hemodialysis, and we actually designed the study to be able to reach the steady state. And as you can see in the next slide, the medication exposure is very similar regardless of the kidney function. We enriched this data with a PK model, and what we find is that kidney function does not play an important role in drug elimination. In other words, the levels of the drug that you get in the blood are very similar regardless of what is your kidney function. So based on these observations, the next slide, we thought that it's worth studying an SGLT2 inhibitor, in our case canagliflozin, in patients on hemodialysis to see whether this can prevent cardiovascular endpoints. However, as there is no evidence with SGLT2 inhibitors in such a low GFR, and the mechanism of action at the cardiovascular level is not fully elucidated, we thought that before designing an ambitious and event-driven study to prevent hard endpoints, a mechanistic phase 2 trial would be appropriate to show if the medication at the cardiovascular level behaves in a very similar way to what is shown in patients with preserved kidney function. So next slide. So we were based on the EMPA-HEART study. This clinical trial assessed the impact of SGLT2 inhibitors on left ventricular hypertrophy using cardiac MRI. And in this placebo-controlled parallel group RCT, the investigators were able to show that empagliflozin reduced left ventricular mass at 6 months compared with placebo. We know that left ventricular hypertrophy is a poor prognostic factor for other cardiovascular endpoints, and we thought that this would be an appropriate endpoint for a phase 2 study. Next slide. So we designed this CARE-MRI clinical trial with the hypothesis that canagliflozin will improve left ventricular hypertrophy in patients with advanced CKD. And this is a placebo-controlled parallel group RCT, and we enroll patients with a GFR below 15, including patients with incident hemodialysis or incident peritoneal dialysis. This is the first 6 months. We enroll only the incident population because there is some evidence that there are structural changes in the myocardium that may not be fully reversible after a certain time point. And also the prevalent dialysis patients, especially those who survive too long, have a very different profile from the average patient who starts dialysis. So we thought that from a mechanistic perspective, it would be more appropriate to enroll only incident patients for such a study. We used some enrichment criteria to be able to have some better some higher probability to identify a beneficial impact, and this is presence of left ventricular hypertrophy at baseline, type 2 diabetes, a cardiovascular event in the last year, and significant albuminuria in those who are not yet on dialysis. And these criteria were very much based on other clinical trials in dialysis, and most importantly, the Alchemist study with spironolactone that was completed recently. And we stratify the randomization by the presence or absence of kidney failure at baseline. Next slide. So how the study works, the patient will be randomized to canagliflozin or placebo. There's no placebo for this medication anymore. The company is not providing us anything, so we're buying the drug from the market. We have a private vendor who encapsulates canagliflozin and in identical capsules put cellulose for the placebo. And then we get an MRI at baseline. We have an MRI expert at McGill, Dr. Matthias Friedrich, who helped very much for the design of this clinical protocol. And we then see the patients in the dialysis unit or in the clinic every 3 months. So we try to minimize the visits. So we have the baseline visit and then the visit at 12 months. And we have an MRI happening at baseline and an MRI happening at 12 months. We also do some other assessments. And we can go to the next slides when we review this with other endpoints. So next slide, please. Yeah. So the primary endpoint is the change in the LV mass to volume ratio from baseline to 12 months. It was a little of debate which endpoint we use to assess left ventricular hypertrophy in dialysis. There was some worries about the potential impact of volume overload, especially in the anuric hemodialysis patients, that would alter the interpretation of the left ventricular hypertrophy. So that's why we used this endpoint as advised by our MRI experts. The other thing we're trying to do is to time a little bit the MRI with the dialysis session and avoid having an MRI at the very end of the large interdialytic interval when the patient can be 5 kilograms above the target weight, and then you have too much noise and it's difficult to interpret. So secondary endpoints are other MRI parameters. Some of them are very standard, such as left ventricular end diastolic volume, left ventricular mass index. But we have also some other techniques. We have the bold MRI sequences. These are MRI sequences where the patient hyperventilates and are supposed to assess oxygenation of the myocardium. It's a quite comprehensive protocol with T1 and T2 maps for fibrosis and for myocardial edema. And we try to elucidate how the medication works, if it works, at the heart level in this population. In addition to this, we are actually looking at clinical endpoints, although we do realize that with our very small sample size, we are certainly underpowered for this. Clinical endpoints are major adverse cardiovascular events, but also for those who are not yet on dialysis, progression to dialysis and residual renal function in those who are already on dialysis. And then we do a 6-minute walk test at the beginning and at 12 months. And we assess quality of life of patients with the Kansas City Cardiomyopathy Questionnaire. And for those who are interested, we are also providing 24-hour blood pressure monitoring at baseline and at 12 months, although this is optional because some patients told us that they find this intervention to be a little bit too aggressive. And then we recalled also adverse events that may be happening, and we have a data safety monitoring board that is reviewing this at regular intervals. So next slide. So the study is funded by Heart and Stroke Grant in Aid. My major problem is recruiting. We wanted to complete recruitment in 2 years and have another year for to complete the study. And this was assuming a 2 patients per month recruitment rate, but we're only at 1.2. There are some practical issues. There is this MRI protocol and a dedicated MRI machine. And there is a little bit limitations in funding, although I don't think money is our big problem here. And the thing is that we have only, this is a single center study. We tried to reach out to other centers in Montreal who could refer the patients just for the baseline, the follow-up MRI. We are in, I will go next week to another hospital. I was a few weeks ago in another hospital, but if there are colleagues from Montreal who are interested in the study, please reach out to me. And the other thing is that maybe we should think more creatively and try to recruit other centers, but that could require a coordination of the MRI protocols across the centers and have somebody who knows much more in detail and in depth about CARE-MRI than I do, about cardiac MRI than I do, to make sure that we're not trying to compare things that may not be exactly the same. Expected study completion, that is probably optimistic, is 2027. So I hope that the study will provide some useful evidence on the efficacy of this medication in patients with stage 5 CKD. Thank you very much.
We have about 5 to 8 minutes for questions, both online and in person. I got one.
You got one?
Right here, do you want to go first? We might have the same one. Let's see.
So I think the notion of doing it in another center, like not Montreal, like if patients are their own controls and the MRIs are followed protocol, then it shouldn't be an issue. You're not comparing, and you can also have them read each other's, right? So you can do another little thing to see that they're getting the same result. So that shouldn't be an issue. And I guess the question is, you've chosen canagliflozin, but there's lots of people starting dialysis that are staying on the drugs that they're on, DAPA, and you could take advantage technically of that, although it's a slightly different question. But many people, like the DAPA study, actually kept people on even when they went on to dialysis. So, and some current practices are keeping people on, especially on peritoneal dialysis. So you might take advantage of doing your extending it in a more pragmatic way.
Yeah, that's a good point that we have to consider. Thank you very much.
Hi, Thomas. Very exciting. I was just curious. This is potentially an idea for a very easy amendment, which is the kidney is right there, below the heart. Last I checked, and I imagine you're using 3T scanners, like MRIs, for this for your study.
For the kidney, you mean? Yeah.
Yeah. I mean, and it may be within your field of view as well. And I know maybe Dr. Friedrich, I know he specializes in the heart, but there are many people that actually very much specialize on the kidney. And if you are using a 3T scanner,
3 Tesla,
there are a lot of established protocols that look at very similar parameters within the kidney as well, including fibrosis, oxygenation, even renal blood flow. And there actually isn't a lot out there. It would be very complementary to your, what you're looking at in the heart. And it's a relatively easy amendment. And from a cost standpoint, you would just need to find the money later to actually do the kidney analyses. And I'm happy
just
to
take the images.
It's just more the images, and it may be worth just making sure that they do some of those protocols. I don't know. It's just a food for thought. I know you probably are well into your study, but with the sample sizes that you have, you may actually be able to get some exploratory data, which would probably be very interesting in your dialysis patients with residual kidney function, to actually look at the kidney and in people with advanced CKD as well. So I'm just
well, that's a very good point indeed. And the only thing is how much time they need to stay in the machine, if it needs to prolong very much, because there are some.
It's not half an hour for the.
It's an additional half an hour, huh? Yeah.
So I don't know unless it's all.
Yeah, I have to ask about this. Yeah, because sometimes slots are issues. It's an issue how many slots you can find for your protocol, because there are other studies using the machine. But that's a very good point. I'll definitely ask. And as you said, it's an easy amendment.
Yeah, and I'm happy to introduce you with our MRI guy as well.
Oh,
Yeah.
thanks very much.
Yeah, amazing study, Thomas. And perhaps this is a naive question, but in pediatric practice and research, we would use echo cardiography to look at LVMI and LVH. Kind of, was that considered? What are the benefits of doing cardiac MRI? Because obviously it increases cost, complexity, concerns you said about
The main
weight gain.
issue is that, it's very much operator dependent, the echo. The good evidence that we have from randomized studies is with MRI and also availability of echocardiogram. It may sound very strange what I'm saying, but it's easier to get a cardiac MRI at the MUHC than an echocardiogram.
Yeah.
Yeah.
Great presentation.
Thank you.
I always love reading your PK work, and going down this line of mechanistic, I think is really important with life cycle going on. My question is, I forgot about LVH sex gender differences I saw in your eligibility criteria. Could you walk me through some other sex and gender considerations for your design and conduct of your trial? If your targeted recruitment of females, if you're looking for any subgroup analysis, even though that it's a small trial, what factors have been considered?
So we're trying to enroll, this population there are more men than women in this population, in the clinics and the dialysis units. And unfortunately, the study sample size is very small to be able to provide some meaningful analysis per sex. And gender, we didn't include that in the design because of the very small sample size. We thought that we will get so few numbers, that will be impossible to draw any conclusion. We're trying to recruit as many women as possible in the study. And at this time, the ratio is probably close to 60-40% at this time. But, it's in a study that's poorly recruiting. At this time, we don't have the luxury to deny
the constant struggle, right?
Unfortunately, yeah.
Were there differences in the PK of canonical flows in by sex? Did you look at that?
So we looked at this in the model. So yes, there is a difference in exposure that you can see between males and females, and that results in different area under the curve. But this is also the case in the general population. So you don't see a different profile in patients with advanced kidney disease compared with those who have preserved renal function.
Time for one last question. Going once, gone. All right.
Thanks very much.
All right. So I have the pleasure to introduce Dr. John Gill. He's a professor of medicine at the University of British Columbia, Division of Nephrology at St. Paul's Hospital, and a research scientist in the Center for Advancing Health Outcomes. He's held national and international leadership roles in organ transplantation and supervised numerous clinical and research trainees. John is an active researcher whose diverse interests include clinical trials, clinical outcomes in kidney transplantation, access to care, health services, and research ethics. He's a recipient of the American Society of Transplantation Established Clinical Investigator Award, the American Association of Kidney Patients Medal of Excellence, the King Charles III Coronation Medal, and is a fellow of the Canadian Academy of Health Sciences. John has done a lot of work in the transplant area and is a great ambassador for us from Canadian nephrology. So please welcome him and me in welcoming him and presenting the PART study.
Thanks, Steve. Welcome to Vancouver. If you were here yesterday, it was a stunning day. It's not raining right now, so thank you for the opportunity. I'm actually supposed to be doing this with Dr. Suri, but I think we're just going to tag team, and Dr. Suri can chirp in from the back room. The reason we're particularly really excited about this study is in transplant in my career, we've seen us become increasingly isolated from nephrology, and this is a study that is actually challenging that, and it's forcing transplant and dialysis doctors to work together. So we really are happy about that. And Rita's been a great partner in helping advance this study. This is a CHR funded study. Oh, next slide. Okay, so these are my disclosures. There's a lot of them, but they're not relevant to this study. And I've benefited, actually, Willig's in the room. I've benefited from support from Astellas Canada for many years. So those are my conflicts. You can go ahead. Okay, so this is a really old study, and I like basic epidemiology that just to set the stage. This is when our patients die, and this is in transplant candidates. And the way to look at this slide is 3 years on the waiting list, first 3 years with a functioning transplant, first 3 years after a transplant fails. These are stratified by age, but the pattern is very obvious. We've focused a lot. In fact, we're doing a trial right now called CARE, which is focusing on this area, the perioperative mortality risk. But in this study, we're talking about this side of things. And this has been a conundrum. It's probably multifactorial, but why we see these high rates of mortality after patients fail. But the role of immunosuppressant in creating this badness has been a vexing problem throughout my career. You can go to the next slide. So as I said, this is about passing the baton, and some of this is in transitions of care between people having a functioning transplant, returning to dialysis. And this is something that I think has been a strength in Canada, but I think is weakening over time. You can go ahead. So a little bit more about epidemiology of transplant failure patients. We don't really think of them as a distinct group, but we really should. So if you count Canada and the United States, there's about 7,000 patients who start dialysis after a failing kidney transplant. And that number is increasing. And that's despite the fact that long-term outcomes in transplant are slowly nicking up. But we just have this huge vintage of people now who have kidneys that are 15, 20, 25 years old that are attritioning and returning to dialysis. And so this population is increasing. So I'd like to say that it's the fifth leading individual cause of dialysis. So if you look in USRDS, they don't actually put it out there. And in CORE, they're not reporting this right now as a distinct group, but it's about the fifth leading cause of starting dialysis. So it's a significant patient population. Go ahead. So historically, this group, despite the fact that they were selected to receive a transplant and generally are younger than our average CKD cohort, their mortality is not great. So their 5-year mortality is around 40%. Leading causes, work with Jeff Pearl and others have looked at access to dialysis access, differences between peritoneal dialysis, hemodialysis, et cetera. So sepsis is definitely a factor. But then there's also this concern about a chronic inflammatory state. Go ahead. And so this is like the obvious graft in the room, right? This was done by my colleague Bruce Kaplan many, many years ago. And it basically was telling the transplant community that your job doesn't stop at the day the graft fails, that we should own the outcomes of these patients after the graft fails. This is just looking at patients with a transplant who continue to function versus those who return to dialysis. You see the huge difference. This data has been replicated. I didn't show you in the interest of time by Greg Knoll in Ottawa. We see the same thing in Canadian data. You can go ahead. This slide requires a little bit of an explanation. And to me, I look at things that are definitely going to work, right? So I'd love to say that SGLT2 inhibitors and all the cardiac things that we're doing, and obviously better access to dialysis and establishing permanent vascular access, those sorts of things are important. But repeat transplantation is for sure going to have an impact on mortality in these patients. And the way to read this slide is patterned after the classic transplant slide that I showed yesterday. The relative risk of one here is either people who are seeking a first transplant who are on the waiting list, or people who failed a transplant and are coming back onto dialysis and are waitlisted. This first curve is incident or first transplant recipients. You can see there's a perioperative risk compared to staying on the waiting list. But then that relative risk of mortality comes down. Very familiar in the first transplant. The important thing is that also holds in people who failed a kidney, right? So people who are coming back to get a second kidney compared to people who are on the waiting list waiting for a second kidney, you still see this survival benefit and relatively similar pattern in terms of perioperative risk and then a subsequent risk reduction. So only to say that repeat transplantation is well justified in these patients. And we could think about that in the context of a resource poor situation. You can go next slide. And this is a slide that just shows that antibodies really are a major barrier to retransplantation. So this shows waiting time, so years on dialysis, and these are people who actually got a transplant, right? So you can think of waiting time for transplant in a number of different ways. This is of the folks that actually got a transplant. How long did they wait? It's not the best indicator because obviously people got delisted or died while they're waiting and so forth. But you can see once your CPRA, which is your level of anti-HLA antibodies compared to those represented in the donor antigen pool, you can see once you go over 95%, your time on dialysis significantly increases, and this is Canadian data. Now, to combat this, we've developed all kinds of fandangled allocation schemes where we're shipping kidneys from Halifax to Vancouver, et cetera. So people will say, well, this really only becomes an issue at the extremes of allosensitization. But I would say that any level of increase in allosensitization is bad. And it's bad because it creates disparities, right? It makes us amend allocation schemes. But importantly, it influences what immunosuppression we give when people do get a second transplant. And so overall, allosensitization, even if it doesn't have these dramatic effects on waiting time, I think it's something that we want to avoid. And we had a lot of discussion with the dialysis teams about saying what should be our primary outcome. And so prevention of allosensitization, as you heard in the trial, in the title, is our primary outcome for the trial that we hope to definitively do. You move on. So to prevent allosensitization, anti-rejection drugs that people took while their transplant was functioning, they continue to take them. And the issue here is there's huge variability in practice. There's a lot of religion about this between sites, even between VGH and St. Paul's. People believe different things. And so only to say there's a huge variability in practice into when people prune immunosuppression, how long they keep it on board, et cetera. And this has been informed only by retrospective studies. And I'm going to show you the only prospective study that's been done on this in a sec. You can keep going. So we do know that if you pull immunosuppression, you're going to get allosensitization. This is a study from a guy named Josh Augustine in Cleveland. He's at Case. And you can see here that if you wean immunosuppression, your odds ratio of getting significant increase in anti-HLA antibodies is huge. So this is probably not extrapolable to all cases, but you can see it's a huge odds ratio after adjustment of at least 14-fold in people who came off immunosuppression versus don't. Of course, there's indication bias in this retrospective study and so forth, but only to say there's good evidence that pulling drugs will crank up your allosensitization. Okay, next. So when we think about this and people coming back to dialysis after a failed transplant, it would be convenient to lump them into two groups: people who are repeat transplant candidates and people who are not. Okay, because our therapeutic goals in those two groups of patients are different. What I would suggest is as doctors, we're not really good at putting people in this not repeat transplant. So we are routinely transplanting septuagenarians who failed a first transplant now. That's a fact. And the day that they come back onto dialysis is not how they look 3 months later. So they improve, and people who we never thought were going to get another transplant end up being considered for transplant. In many cases, if they have access to transplant, we would transplant them again. But in repeat transplant candidates, our goals really are to prevent allosensitization. That's our primary goal. And so continuing immunosuppression is the goal in those people. The people who are not repeat transplant candidates, we don't want the failed transplant to cause rejection or symptoms, which it can do after it fails. So the goal is to, as quickly as possible, wean off that immunosuppression because we know they're not going to come back for a transplant. So we don't really care if they get allosensitized. We just don't want them to have clinical rejection of the transplanted kidney, which is called graft intolerance syndrome. It can lead to a whole bunch of issues, including increased cardiovascular risk and increased inflammatory state, et cetera, et cetera. In the mix, of course, is you've got the complications of immunosuppression. So remember, these are basically the same drugs that these patients were taking for years before their transplant failed. So there's the cumulative toxicity of that, but then there's also whether the side effects of those drugs is accentuated when you put somebody on dialysis and they're having blood taken out of their body and passed through a foreign substance and put back to them, in simple terms. So this leads to sort of a personalized thing, but I would just argue that this is sometimes hard to implement for the reasons that I've outlined. You can go to the next slide. So these are opinion-based recommendations of both the American Society of Transplantation and the British Transplant Society. And basically, if you fail to transplant, obviously, if they have symptoms, then we rarely have to take the graft out, right? So taking a failed transplant out is not a small endeavor. It's a bloody, difficult procedure. There's often scarring and stuff. And so the bottom line is it's not the preferred thing unless our hand is forced. In the absence of symptoms, it really comes down to whether you can dichotomize patients into whether they are or are not a repeat transplant candidate. And then you can see here that these recommendations are not really satisfactory because waiting times for transplantation are far greater than 1 year. So this is kind of waffling based on the fact that we don't really have great data, but that's what's out there in terms of what we should be doing in patients. Next slide. So all the data on this was retrospective. So Greg Noll and I did a study that was published a couple of years ago back now in JASN, and it challenged a lot of what we think we're doing in this patient population. So the goal here was to do a prospective study, not to dictate what people did, but just basically to prospectively see when drugs were weaned, when people got hospitalizations, when they died. Take it forward. And this is multi-center Canadian CHR funded observational study. So the first thing is we continue drugs a lot longer than 12 months, right? So the opinion race recommendation, everybody would be off and would be blank over here. And you can see that we continue drugs for a lot more than 12 months. You can see the CNIs are the most likely thing. We also continue prednisone a fair amount. It's continued. And the anti-metabolites, surprisingly, are even continued out to 36 months. So in Canada, we're leaving a lot of people on drugs when they've got a failed kidney transplant. Next slide. So what happened in this? There was 269 patients in these centers, in all these centers, and you can see that we had a significant mortality rate, lower than what had been previously reported. I don't know if that was just a protective effect of being in the study, but in any case, we had significant mortality. Next slide. And the other thing that was really prominent is we had a lot more hospitalization events, 143 cases. And so continuation of immunosuppression was not associated with a higher risk of hospitalized infection. You can see that the hazard ratio here is 1.81. So it definitely was it definitely is not out of the realm that it was possibly associated with the first infection. But you can see in terms of multiple infections, it didn't really come out. So the overwhelming thing here is it's hard to tease out this as the causative factor that's driving sepsis in this. Next slide. What about allosensitization? So the class 1 and class 2, this is how we group anti-HLA antibodies, did go up in these patients. And you can see that they did go up significantly in this relatively short period of time, even though the fact that we're continuing immunosuppression. But the interesting thing is it didn't differ by those who continued immunosuppression or not. So you can go to the next slide. So you can see the class 1 and class 2 antibodies, and it's grouped in the blue, the ones that stayed on, and the red are the ones that stayed off. Now, the problem with an observational study, as you know, is we're not really rigorously making sure patients take their medications. We're not doing CNI levels. So one of the limitations of this observational study is are our patients actually doing what we think they're doing? How am I doing for time? Oh, I'm okay. Okay. Well, next. So, and then there was some people who had symptoms of rejection in the failed graft. There was 21 events in 18 patients, but we didn't find an association with immunosuppression weaning or not. Next. So these challenges, I think, challenged the current paradigm. Drugs were on board for longer than a year. The drugs there wasn't a smoking gun in terms of associated with badness in terms of mortality or hospitalized infection. And the drugs, shockingly didn't prevent allosensitization. And again, we have some limitations with this observational data because we don't know how rigorously people were taking meds. Next. I just said all that stuff. We can go on. So this brought us to where we're at now, which is the PART study. So this is me and Adeera Suri thinking this is what we look together when you amalgamate us by AI. I don't know. Next slide. So we are doing an RCT, so as to say, we are in the midst of, and so the hypothesis here is that continuation of immunosuppressant drugs versus withdrawal will be associated with a lower risk of a clinically relevant increase in allosensitization. And so I can briefly define that. Basically, we're saying if they change their antibody level by a level that will cut their eligibility for transplant by 50%, we're calling that clinically significant. Okay, so if you start at 0, that means your CPRA goes to 50%. Sorry. If you start at 80%, that means you would reach this outcome if your PRA went to 90%. Okay, in simple terms. So the definitive trial will also evaluate the use of a novel biomarker, which is how well matched that kidney is to the donor. Now, it's interesting, right? When we do a transplant, we do all kinds of compatibility testing, but we don't do that when the transplant fails. So one of the things that could be quite important here is using this as a biomarker. So eplet mismatching, I'm not going to go into big detail, but it's like HLA matching, only much, much more sophisticated in simple terms. So whether that is something, so we're going to be collecting that in the background to see if that has anything to do with the price of eggs. Keep going. So this is what we would be telling patients as they're going on to the, and so I know there's some patient representatives, but this is the highlights of what you would hear. We know that there's a yin and yang with potentially continuing immunosuppressants. Our primary goal is to maintain access to a repeat transplant, and this study is determining whether weaning those immunosuppressions will actually prevent you from developing antibodies while not increasing a risk of badness, infection, cardiovascular disease. So that's the script. I decided to throw that in. Next. So right now we are funded to do a pilot in 8 Canadian cities, and this is to demonstrate that we can recruit patients, and more importantly, that investigators like Lakshman Ganaratham will actually wean the immunosuppressants into people. This is obviously an open label study, or will they chicken out? And it's interesting, people wean immunosuppression willy-nilly all the time, but now that we're doing it in the context of study, everybody's freaked out about doing it. So anyways, keep going. So what are the inclusion criteria? Pretty broad. This is an adult study. You can't have an extrarenal transplant. You have to be on CNIs, which is like 95% of our patients. Obviously, you can't have a reason to take immunosuppression off, like a cancer or an infection. If you're HLA identical, this is a non-starter for you. If you don't have samples that we can determine this biomarker of eplet mismatch, we don't want you in the study, but we're working around that. And then if you failed your kidney really quickly and we think you're at really high risk of rejecting that failed kidney, we don't want you in the study for safety reasons. And then if you're a non-transplant candidate, like if you've got an active cancer, you really got limited life expectancy, we wouldn't want you in the trial. So it's pretty broad. The exclusions are fairly sensical. Next slide. This is what happens. So basically, people coming in, we're going to recruit them before they actually hit dialysis. We're going to determine their baseline level of allosensitization. We're going to determine their eplet mismatch, and then they're going to be randomized to continue drugs for 2 years or to wean off by 6 months. And really, the people that are going to continue drugs are primarily going to maintain CNIs, and we're going to measure drug levels in this. In addition, we are measuring things like residual renal function. We do uniquely have a significant peritoneal dialysis after transplant failure population, where that's going to be an important outcome. Go on. This just shows you how we're pulling the drugs off. I don't think we have to go into detail. Basically, you're going to come off of everything at 6 months, or you're going to stay on. In people, there's about 30% of our patients who are not on steroids. So our center is one that doesn't maintain people on steroids. What are we going to do in those people? So if you're not on them, we're not going to start them if you get randomized to continue immunosuppression. You'll just do whatever you were doing. So if you're on them, you're going to continue. So there'll be a little bit of heterogeneity with regards to prednisone. There is some concern that weaning prednisone in those people that are on, that'll come off, that they may have hypoadrenalism. So there are some real issues with this. Okay, next. Okay, that's the end. That's good. So transplant failure patients, 5th leading cause of starting dialysis. It is a big deal. They don't do well. Got to transplant them again if you hope to change things durably. And in repeat transplant candidates, how we present allosensitization. And this is primarily focused on CNI, so we could talk about which drugs and why we didn't include that in the design. But that's what we're after here. Thanks for your attention.
Okay, we have about 5 minutes for questions. Put the microphone over to John, congratulations on getting this study funded and getting it started. I have 2 questions. So with regard to the epitope mismatch, why would you expect it to be surprising when we know very well the degree of epitope mismatch with the person with the transplant or going for a transplant does increase the risk of alloantibody development, rejection, and so forth? Why would you expect anything different?
Yeah, I don't. I actually expect it to be the same, Lakshman, but I do think that it's an expensive test. And so if we're going to do it, having the data to back it up And if we're going to change paradigm to say, look, when a transplant fails, we should be going back and determining the eplet mismatch, and that may inform the speed with which we withdraw immunosuppression. I think having the data, so to change practice, as you know, you got to have data, right? So that's the reason. I don't expect it to be different.
Thank you. One other quick question. Is that okay, David? Okay. Second one is about the types of immunosuppression. There's a lot of controversy, mTOR versus CNI. So here you're taking an all or nothing approach where you're completely off or not. Can you comment on the type of immunosuppression and how it may affect allosensitization?
Yeah. So what we learned during COVID is that mycophenolate is definitely important in terms of antibody response, probably more so than CNIs, right? But there's a practical issue. If you look at current practice patterns, most people are pulling anti-metabolites, and there's a good reason for that. The anti-metabolites accumulate in renal failure, right? So they're not well tolerated. So unless we're putting people onto azathioprine and so forth, getting them off of MPA-based drugs, we're going to have a problem. And so in both arms, they're going to come off, both arms are going to come off mycophenolate. So that'll be the same. And really, if we look at what's going to happen is one group's going to stay on CNIs and the other group's going to come off. So mTOR is really small potatoes in terms of our use in Canada. Last time I looked, we're looking at about 2 to 3% of patients being maintained on mTOR inhibitors. So it's really a small potatoes thing. Does that answer your question? Yeah. Go ahead, Dave.
Yeah. So I'm really interested. So not a transplant nephrologist, but I know what an eplet is because I trained in Winnipeg. My question is about your outcome and the fact that your baseline CPRA could be all over the place. So that absolute risk at baseline is variable, and informing your effect sizes is also from observational data that you had a lot of biases. Could you tell me a little bit about how you're monitoring an interim analysis or approach to defining whether you're in the right ballpark for your event rate?
Yeah, good question. So right now there's only observational data in terms of from our primary study, the observational study that I showed you, David. So when we track those changes, taking into account where people started, for a definitive trial, we've estimated, and it took a fair amount of math to estimate this, that we're going to need about 800 patients. So you're absolutely right. But the interesting thing is the percentage change in CPRA you would think was related to where you started. So somebody coming in with, let's say, a CPRA of 0 versus a CPRA of 30 at the time they started, you would think that the guy with 30 would be more twitchy. We did not see that. So it's really interesting in terms of that. So I think it really depends on what happens after the fact and when you got that CPRA. So it doesn't include, though, David, however, the change from the time you got your first transplant to now you're failing. We're only looking at, so it made the people that were, let's say, 0 and now 30 at the time they fail, they may be different than the people who were 30 the whole time. I don't know. But so your point's well taken. Right now, based on that data that we had from the observational study in 270 patients, we're looking at an 800-person definitive trial. So this pilot's 96.
I look forward to working with the transplant physicians at my center to help with that number in the future.
Okay, thanks.
Okay, I think that's that for the last question. So thanks so much, Dr. Gill.
Me again. And I am pleased to introduce one of our last speakers, Dr. Istvan Mucsi, a clinician, investigator, and transplant nephrologist at the Multi-Organ Transplant Program and Division of Nephrology at the University Health Network, UHN, in Toronto. He is also an associate professor of medicine at the University of Toronto, and his research focuses on understanding and reducing barriers to kidney transplantation, particularly among racialized communities, and understanding how stress and mental health affect transplant outcomes. Dr. Mucsi is also a principal investigator in the Kidney Health Education and Research Group leading initiatives to improve access to care for underserved populations. Very noble. So I introduce Istvan Mucsi. Oh, he's online. Sorry about that. I didn't know.
Yeah, good afternoon or morning, everybody. I'm Istvan Mucsi, and I'm sorry I couldn't be there in person. I'm talking to you from Israel, where I'm attending the Israeli Society of Nephrology meeting. And today I will be talking about this second part of interest of mine is focusing on physical and mental health symptoms in patients with chronic kidney disease at various stages of their kidney disease journey, and assessing and managing those symptoms. I thank for the organizer to allow me to talk about this. This is my disclosures. I had some grant support from Stellus and Paladin. Today I will briefly talk about the significance of symptoms and how to assess them using patient-reported outcome measures, PROMS, which are questionnaires. We'll talk about the study, and within that part of the talk, I will highlight some of the elements of the study that we did, and also what we hope to enlarge into a larger trial in the future. So I don't really have to spend too much time on this slide, although it's very busy and complex, but people in the room and online know how frequent and how bothersome the different physical and mental health symptoms are for patients with different stages of chronic kidney disease. These figures are taken from a recent meta-analysis, and just to navigate you on this busy slide. So this panel shows the frequency of symptoms in patients before starting dialysis. The middle panel shows the symptom frequencies in patients who are on dialysis, and the right panel shows the same for patients who are transplant recipients. And these probably invisible words are the symptoms that we talk about. And these vertical lines here in these panels represent 50% frequency. So whenever these dots are right to the 50% line, it means that more than half of the patients suffer from those symptoms. And what are those symptoms? As you know, these are diverse symptoms. These include fatigue, chronic pain, itch, gastrointestinal symptoms, poor physical function, and sleep issues, amongst others. And these red arrows indicate where these symptoms are in terms of the hierarchy. And as you see that many patients, both before, during, and after dialysis initiation and after transplant, suffer from these. And these symptoms have a significant impact on quality of life overall, again, amongst other effects. I speak for myself. When I work with patients in the clinic, I usually focus on kidney function, immunosuppressive treatment, and managing some other conditions. And I don't really think specifically on about the symptoms. We tend to think that when we treat the kidney-related issues, the symptoms will somehow automatically get better. Frequently, however, this is not the case, and specific symptom management efforts would be needed, which we know that can work and can improve symptom experience and overall quality of life in patients with other conditions, including cancer. And how this is done is frequently done using patient-reported outcome measures, which are questionnaires. These are more recently quite precise measurement tools that assess these symptoms that patients will experience. And these tools can, similarly to the hemoglobin measurement or the potassium measurements from the blood, the measurement of the symptom severity could inform our treatments and management of those symptoms. Now, this has been tried, as I said, in patients with cancer and in some other clinical conditions. And of course, it has been considered in patients with kidney disease as well. This first study that I will mention here is from Canada. This is the Empathy trial that used a set of questionnaires, patient-reported outcome measures, to assess symptoms in patients on dialysis, mainly in Alberta, with in regular intervals. There was quite a detailed plan what to do with the results, and the clinical team, the dialysis nurses, were trained to monitor and address those issues. And the hope was that this would improve communication about symptoms between patients and clinicians. And unfortunately, this trial did not confirm this, and it was negative. There was no improvement. And the reason behind this is somewhat unknown or complex, but potentially includes the fact that in spite of all the education that happened before the trial, neither the clinicians nor the patients fully understood what the questionnaires were used for and what the purpose of this project was. Nevertheless, currently there are at least 2 large trials, one in Australia, one in the UK, using a very similar design where symptoms would be assessed using questionnaires at regular intervals, and a predefined symptom management approach would be applied if the symptoms were significant. And the outcomes would be mainly quality of life related outcomes in these trials. So on this background, we have been working with patient-reported outcome measures and validated a number of these tools. And more recently, we were able to complete a pilot study, which we called support dialysis study. And this was to assess whether a large trial using PROMS systematically to inform symptom management efforts in patients on in-center hemodialysis would be feasible. We wanted to find out if this trial could be done. Accordingly, the objectives and the hypothesis for this study was that we thought that a larger trial would be feasible, and we wanted to demonstrate that using specific feasibility outcomes. And we also were hoping that the study intervention and the procedures would be acceptable both for patients and clinicians. These were the criteria that we defined for the feasibility assessment. We were hoping that we would be able to recruit patients within a 3-month period into this study. We planned a 60 patient recruitment. We hope that the patients who consented to the study would stay in the study. We were hoping that the majority of the patients whom we asked to participate would join the study and they would not drop out. And of course, we collected a lot of other data, both questionnaires, PROM data, and clinical data, which reflects the plan for the subsequent study data collection. The design overall was a parallel arm, non-randomized study design. This had some practical consideration behind it. The intervention arm was at the Toronto General Hospital because at that time when we were planning the study, some patient-reported outcome measures were already integrated in the electronic patient record, and the control site was at a different hospital. Inclusion was quite broad. Any prevalent patient on dialysis, in-center hemodialysis, was eligible if they spoke English, because the questionnaires were in English, and if they did not have any planned interruption to their dialysis treatment, which would be transferred to PD or transplant. We spent about 2 months in kind of site preparation where we provided information both for patients and clinicians about the overall study and the purpose, about the significant symptoms, about the symptom management, symptom assessment tools, and also how to manage those symptoms. And after that, we had a 3-month recruitment period, and subsequently for 6 months, symptoms were assessed monthly. And if the patient had mild symptoms based on or no symptoms, then the clinical team was supposed to support patients with the management of those symptoms. If moderate or severe symptoms were detected, then we contacted the clinical team to suggest a referral to different specialty clinics that could address those specific symptoms, and we'll come back to that later. So as I described here, the study intervention actually had 2 components. One was the assessment of the symptoms using PROMS, the questionnaires, and the intervention part, which was supporting self-management and also referring to specialty clinic. And I wanted to highlight a couple of the components in the study. One was the assessment, where we used patient-reported outcome measures developed by the PROMS program in the US with significant NIH support. And the reason we did this, these are highly validated, precise questionnaires that were shown to be valid in multiple clinical populations. They assess some of the frequent symptoms that are available or that are present in patients with kidney disease. And they also provide the opportunity for this computer adaptive testing, which is a new way to assess and measure symptoms or functions. Of this using this chart and using the example of physical functioning. If we want to measure physical function in a large number of people, some of them will have poor functioning, some of them will have very high functioning, and the majority will be in the middle. But in order to assess both the low, high, and the middle function, we need to have questions in the questionnaire that address each of these levels of functioning. And this would include a lot of questions. Computer adaptive testing will result in a different process whereby the algorithm selects the same questions for everybody. But if we want to assess the function of this, the response will place them on this range of the scale, and the subsequent questions administered by the algorithm will focus around that level of function where the person is, and this prevents irrelevant questions being administered, and also it will result in a very brief and very precise assessment. This set of questionnaire is very responsive. These curves represent the changes after transplant in incident transplant recipients in physical functioning, anxiety, and pain. And as you see, within 8 weeks, the scores reflect the changes that patients experience during recovery. And this set of tools was used in the recent COMBIN study as well, which again demonstrates responsiveness and sensitivity. We created scorecards for the patients to visually represent the scores. And when the patients had significant symptoms, in the electronic version of the scorecard, there was a hyperlink linking the symptom to our symptom care website, which we built. Here, patients could read information about the nature of the symptom and also receive self-management support. Integrating this into health records was an important feature which we couldn't achieve, but hopefully this will be a way of facilitating use of these tools in the future. And finally, the assessment in our program had an important difference between previous studies. As mentioned, in the previous studies, including empathy, symptom management was left for the clinical team, for the dialysis team. And there were some issues whether the dialysis team was well equipped to do this. And we were lucky to have a specific renal supportive care team that was manned by a couple of palliative care physicians and fellows and the social worker. And this model of care has been shown in Australia and has been increasingly used in the United States as well to specifically address the symptom burden in patients with both pre-dialysis, dialysis, and transplant, kidney transplant population. And of course, this can be also important in comfort-focused care. So with this, we were able to achieve recruitment, retention, and had a low dropout rate in the study. So most of the feasibility criteria were accomplished. The completion rate of the questionnaires was somewhat lower than we expected, mainly in the control arm, not in the intervention arm. Acceptability was also reasonable. At this point, in conclusion, we think that this approach may be useful with modification after the learnings from the study. And hopefully we can extend it and plan together with patients a larger trial. And at the end, I would like to acknowledge my funding resources and our partners. Thank you very much for the attention, and I'm happy to answer questions.
Thank you so much. We'll open it up to questions. We have about 3 to 4 minutes. I got one. No kidding. So I'm a symptom guy, and I do trials in this area, and I think this is very important work to address patient-reported outcomes. My question is about selection of your sites for your definitive trial. I think, handing the baton to clinicians that are comfortable managing symptoms, and not everyone, I'll speak about my experience in Edmonton, has a sleep clinic that they can refer to, or a diverse group of palliative care physicians that will see renal patients. Could you tell me, and that was one of the risks with the Alberta study, right? So I'm talking a little bit about from own experience, but how are you going to select your sites so that you have heterogeneity outside of UHN and are selecting a diverse group of patients, including from community settings?
Yeah, thank you very much. Certainly, this the management arm of the intervention is critical. And at this point, although those specialty clinics are important, but most of the symptoms can be managed on a more general level. And this is where I think the so-called renal supportive care approach may be a good intermediate. In a number of our community partners whom we have been working on the validation side, they indicated that their palliative care team is quite interested in collaborating in such efforts. So I would assume, and this is what has been happening in the United States as well, that palliative care physicians may be helpful in this endeavor, joining the nephrology team, because leaving this symptom management entirely for the nephrology or the dialysis team seems to be challenging, I would say.
Thanks so much. Are there any other questions from the room or online? I don't think so. Thanks so much, Dr. Mucsi.
Thank you very much.
All right. All right. Can we go back to the slides? So thank you to everyone to sticking it out to the end. We saw a lot of great work going across Canada in the transplant space, in the dialysis space, in the CKD space with multiple groups of investigators. So lots to be excited about. We're going to just highlight a quick mention about some CNTN resources for people available. And before I end, I just want to make sure, can people just stick around so we can take a photo? It would be great to update the website post-COVID with a group of people that are engaged and, interested in clinical trials. So just, if you need support from CNTN, a lot of people have utilized our peer review services, letters of support, review of grants and response reviews, etc. Reach out to us. We're happy to help from a scientific operations committee. There's lots of work that we can assist you with. So contact myself, contact Hans, contact Omo. All this information is located on our website. There are timelines, so get to us in advance. We also offer French review too. So it just really depends on what you're looking for. A quick shout out to Kidney Link as well, poster study so that people are aware of it. Lots of more exciting work going on that wasn't presented today. And then lastly, there's also our peer review for patient partners. Next slide. And then thank you so much. Feel free to stick around, network. I know people are really busy, but let's try to get that photo in. And thank you again to Omo for all of her hard work, Cynthia, Amanda, Heather from Cancel CKD, and everyone for coming today. Take care. Okay, photo. Don't run away. Yeah. So maybe you want to okay. We'll get everyone to come to this side of the room if you can. More people in the front. Okay, we're just going to go along this wall here. And it's going to come from this way. So if people can just line up this way, and we'll pull this a little bit out. Okay, so just let's fill in here. White can go in the back. Yes. If anyone needs a chair, it's a lineup for okay, we can say that. There's one photo where it's just my eyes peeking through everything. Okay, I'll say it. Can you do me no. Sorry. Is that okay like that? Okay. There can be an empty chair next to me. There you go. I'm over here. Can Arlene, will you come forward? Okay. Kathy, we have a seat for you right here. Background. Okay, thank you, everyone. That means strategically. I'm just going to take a couple of
I don't have much. I'm just